So one of the things we realized when we started talking with teachers about CRISPR, we were very anxious to just talk with them about how this Cas9 enzyme is used as a tool by molecular biologists to edit a human gene. But of course, we learned very, very soon that teachers are really interested in viruses, and they want to be able to talk to their kids about what is a virus. Even before you get to coronavirus, they want to talk about viruses in general. So, when we just casually mention that CRISPR is an adaptive immunity system in bacteria, um, it was that protects the bacteria against viruses, against bacterial viruses known as bacteriophages. Then that really caught the teacher's interest. So we backed up at that point then and started thinking about how do you teach CRISPR biology. So here's what we call the CRISPR Adaptive Immunity Kit that you could use to explain to your students how this system works in bacteria. Up here we have 3 bacteriophages. And we know they're different because you can see that the double-stranded DNA genome contained by each one of these viruses is colored a different color. And this is the bacterial cell membrane. So these guys are on the outside, and they're about to try to infect the cell by introducing this double-stranded viral DNA into this cell. So this virus here with the purple genome is settling down on this membrane, and in a complicated molecular process, it's going to inject its DNA into this cell. Now, we can see here that in fact, this bacterial cell has had a previous run-in with this particular purple virus. And we know that because in a particular section, a small section of this bacterial genome, there's something that's called the CRISPR array. And it's a sequence of genes that people over the last 30 years have come to recognize as being diagnostic of, a CRISPR system. This bacteria has a CRISPR system. Not all bacteria have CRISPR systems, only about 50% of them. So that's another side question that we could talk about if we had more time. But this bacteria has survived a previous attempt by this purple virus to infect it and kill it. And it survived because it was able to capture a small fragment of this genome and incorporate it into this CRISPR array. And then from this CRISPR array, deploy a series of Cas9 proteins programmed with some RNA that makes it possible for this Cas9 to provide constant surveillance for the entry of this purple bacteriophage genome into the bacteria again. And I just said a lot in the last 30 seconds. So let me go back and try to go through that more slowly then. So as an adaptive immune system, we're going to adapt to a prior exposure to the purple virus by deploying a bunch of enzymes that we're not dealing with here in this very schematic, simplified view of CRISPR. But we're going to send some enzymes out to cut up this viral genome, this purple genome, into pieces. And then we're going to bring one piece back and we're going to integrate it into this CRISPR array in the bacterial genome, just between these 2 black diamonds here. And let me stop right now and let's look at this CRISPR array. This is what that Japanese group discovered and reported in the sequence that was just downstream from an E. coli gene that they were sequencing. What they reported were some short palindromic repeats. See right here, short palindromic repeats. They noticed these sequences, which were about 30 nucleotides long, and they were clustered together. That's where the clustered comes from. But then they were regularly interspersed with these different colored sequences. The different colored sequences were also the same length, about 29 to 30 nucleotides long. So they were regularly interspersed with the same size of a sequence, but these were different sequences. These short palindromic repeats were almost identical, which is what called their attention to this unusual sequence in the first place. All right, what we know now, after many, many different people looking at this and having ideas as to what this might represent, what people finally recognized is that, the sequence that is interspersed between the short palindromic repeats, these are viral sequences, or sequences from other pathogens that are trying to infect this bacteria. So that was really the whole key then, that, this might be an adaptive immune system, that you express or you transcribe the Cas9 gene, which is this white gene. You make a piece of messenger RNA, which is then translated by a ribosome to give you these 3 proteins, Cas9 proteins. Okay? Now, notice that this gene for Cas9 was transcribed into messenger RNA and translated into protein. These other genes were also transcribed by an RNA polymerase into RNA, but these are not messenger RNAs, these are structural RNAs. And what I've just said is that the tracer RNA is the name we give to this orange piece. The tracer RNA is complementary in sequence to a portion of the short palindromic repeat, and then that dual RNA is bound by a Cas9 protein. And the key here is that this RNA is going to program this particular Cas9 to look for a purple bacteriophage DNA sequence, while at the same time you're going to form this dual guide RNA that's going to associate with another Cas9 enzyme. And this one is programmed to now search for a yellow bacteriophage genome, and similarly. So, depending upon this bacteria's previous exposure to viruses, it's going to deploy a whole series of RNA-programmed Cas9 enzymes that will constantly surveil the inside of this bacteriophage on the lookout for different, I didn't mean bacteriophage, constant surveillance of the bacteria for the introduction of bacteriophage sequences. And when these Cas9 enzymes find that particular sequence, it's going to cut it and inactivate that bacteriophage genome. So that's the adaptive immunity system.